just for sitting still for so long and then like getting to like like we're
Oh, yeah.
We stop the stream after we eat and we'll record it. The next one, like we'll record it shortly after.
Yeah. We're good.
It's not showing up there?
saw that.
Oh.
So this is, we went to the Ability Expo that was right there in Schaumburg. So this is a company from Poland, Pragma Systems. This is not what we see at the clinic here. So they have the cages. Yes, that's where they do the bunching. So this Pragma system, it's like whatever. She's trying to wait there. She's like, she doesn't want to because she doesn't like to wait there through her legs. But this is like making her where she's pretty much standing.
So it's a good mix up.
She can do like, that's what you want with that.
Yeah.
And then our starting
I can see
point
like
for
trying to
this.
get this
To
jig.
United States, we have like salespeople.
But yeah, they made it right. And then made it real right. You're like, oh my God, that's big.
Keep, we're good. It's going now.
Like seeing how much equipment here costs.
Yeah.
And you can get it installed in your home. It's not really big. It's just,
Yes, you do.
and it comes with like all these different attachments. So you can jump.
Yeah.
So we're actually building, like we're doing a build out now at the house, so she can have like her own bedroom in the future when she gets older, like she does have a big bedroom with
Testing.
with her own bathroom and then all that.
That's cool. Yeah.
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 Okay, how do we look into it? Like I always thought it's learning as you know. That's not. Yeah.
I read some friends Instagram the other day. It's like, it doesn't matter. People are just like, they're not different. And they do. It's like, we're doing it too. You're like, their dog is like the inner.
Okay, we're gonna get started.
Okay.
So, in addition to the people here in the room, we're broadcasting on Facebook Live. So hi to everyone who's joining us remotely today for this part of our day. So, and welcome to everybody here who's in the room. So we spent the morning kind of in informal groups talking about different aspects of read disorder. And now we're moving on to the more formal part of the day. And this is kind of our first test run of a meet and greet session. We've got sessions planned in September in San Francisco and LA, and in I think November in DC, in February in London, England, and in March in Toronto. So, so this is the first one. So thanks for being part of this. Okay, so I'm going to start by talking about. I think this session is called Path to Treatments and Cures. So I'm going to talk about my son Bryson and about Cure Grin. We're going to talk a little bit about some of the basic GRI science, and then talk about the sort of treatments and cures, and then a few minutes at the end for questions. So this is my family. This is a couple years old. So Bryson's there in the middle. And so when Bryson was about 3 weeks old, we went to get baby pictures taken. And the photographer was trying to get Bryson to do a shot where the kids kind of like prop up their heads on their fists. And Bryson's head just kept flopping over. It wouldn't stay on his fists. And the photographer had never seen this with anyone else. And this was really the first indication we had that Bryson wasn't developing typically. And so at his 3-month checkup or 2-month checkup maybe, we mentioned this to the doctors and they confirmed that yes, he had low muscle tone, hypotonia, and he also wasn't tracking objects properly with his eyes. So those were the initial diagnoses. And then as he got older, he got diagnosed with global developmental delay. So he's a happy 16-year-old with his missense mutation in the GRI1 gene, severe cognitive and physical disabilities. He doesn't have epilepsy like many of the kids. And Unlocking Bryson's Brain is a podcast that I did with the Canadian Broadcasting Corporation about our family's search, first for a diagnosis and then for a cure for Bryson. The audio's not playing, so I'm going to skip that. So this is a bit of a timeline with our road to cure GRIN. So it wasn't until 2015, when Bryson was almost 10, that he got a diagnosis of GRIN1 through whole exome sequencing. The next year, we traveled to connect with other GRIN1 families. So first it was in Colorado, and then Pittsburgh, and then we had a meeting in 2018 in Toronto. And that's where we met some local researchers in Toronto that kind of offered really some hope. And that's when I more really decided that I wanted, and it was really important to do whatever I could to try and find treatments and a cure for Bryson. So I'll talk about that hope in a little bit, but that all started in 2019, and that's what led to the podcast and led me to get involved with a group of parents that were coming together to form what became Cure GRIN. And so since Cure GRIN was formed, I won't go over all of this, but in 2020 we got a $600,000 grant from 10 Zuckerberg Initiative, which was just incredible for us in terms of being able to grow. We couldn't use those funds on research, but we did lots of fundraising with families and have been able to raise a lot of money for research and other programs. And then here we are in 2023 with our Chicago meetup in Niles, and that's the Niles leaning tower, the leaning tower of Niles. World famous. It's half the size of the one in Italy. So some of the key milestones for Cure GRIN, I mentioned 10 Zuckerberg. We made a decision to expand beyond GRIN genes to also include GRIA, GRIX, and GRIDS. And we've had we have meetings every 2 months now where we invite researchers called the GRI Genes Roundtable. And we've had over 200 different researchers that have been part of those calls, either as speakers or as listeners. And one of our speakers, Dr. Lonnie Walmuth, has been a regular there. We'll hear from him later. We developed the research roadmap that I'll talk more about. So far, we funded $637,000 in research and done other things to attract more researcher funding. We launched Reconnect, and then we've applied to get an ICD-10 code with GRI Foundation and others, and waiting on the results for that. So I've actually applied for 9 different ICD-10 codes for the different genes where we have families in our group. And our mission is to improve the lives of people around the world with GRI disorder and their families. And really what our key reason for being is to drive patient-centered research that will lead to treatments and cures. Sorry, guys. So we're saying we want to find treatments and cures by the end of the decade. We also run programs that unite and empower the global GRI community, and then strengthen our operations. A lot of that has to do with the fundraising that we need to do, the development, and trying to get grants and sponsorships and family fundraisers that can help us achieve those first 2 things. And it's not just us alone, so there's groups around the world. GRI Foundation is president is Marcia Ashes here today in the room, and there's organizations in Europe and Israel, Brazil, and other places. And so we are not in this alone. We work and appreciate the work of all of the other organizations. So a little bit about GRI disorder. We're going to hear more about this today from the other researchers. But what is GRI? So many of you might know GRIN1 or GRIN2B or GRIA2, but specifically GRI stands for glutamate ionotropic receptors. And these are a form of receptors in the brain. We'll talk a bit more about those. And there's 4 different receptors that are coded by these genes. So the AMPA, NMDA, K-nite, and delta receptors. And so when you hear the name GRIN, it's that GRI, that glutamate receptor ionotropic, and then the N from NMDA that makes it GRIN disorder. So how rare is rare? We know these are rare diseases, but GRIN disorders are actually on the more common side of the rare spectrum. So if you look at some of the more common ones listed here, GRIN 2B is 6 in 100,000, GRIN 1, 5 in 100,000. And in total, if you add these all up, it's 32 per 100,000. And so if you compare that to much more common diseases, Rett syndrome or Dravet's, which all neurologists know about and know a lot about, combined the GRI disorders are larger, but because they've been identified fairly recently as monogenic disease-causing genes, meaning just one gene is responsible for the disease. Not as many doctors know as much about it. So common symptoms of GRI, it really varies by gene, it varies by person and variant. By common intellectual disabilities, speech impairment, gross and fine motor disabilities. Somewhere around half of patients have epilepsy, but that's going to depend on the gene. And so there's a number of other symptoms on the list. Depending on the person, they'll have different versions of these. Okay, so I said we'll talk a bit more about what the receptors are. So we start with the brain, and within the brain we have neurons. So neurons are basically one type of cell that exists in the brain. There's others as well. And so this is a picture of a neuron there, and then if you see in the upper right, it says synaptic or synaptic terminals. And so on those synapses that are part of the cell, and Lonnie, Dr. Walmuth will correct me if I'm simplifying this too much and getting any of the science wrong, but on those synapses are where we find the receptors. So we have millions of neurons, billions of synapses, and even more receptors on those synapses in the brain. And so 3 of them are listed here, the AMPA, the kainate, and the NMDA receptors. And when I say cells that fire together, wire together. So what that's about is there was a researcher, Donald Webb, who kind of was first to recognize that that when we're learning things, what actually happens is certain neurons will speak to each other. And basically what this means is that the more those neurons, the more times those neurons speak to each other, a connection gets stronger and they kind of get wired together. So basically, like, as you're learning about what I'm talking about today, the first time, maybe you understand a bit of it. The next time you'll understand more. And we retain knowledge the more times we hear something or the more times we do something. This library where we're holding this today, maybe if you came back in 2 years, you wouldn't remember where this room was in the library. But if you came back tomorrow and the next day and did that, those wires would become stronger and you'd probably do that. And you'd probably have a better chance of remembering. And the reason I mention all this is that these receptors play a really important role in how neurons talk to each other. And so for our children, one of the deficits that happens with GRI disorders is that when neurons can't speak as well to each other, one of the things that gets impacted is learning them. And so why does it happen? So this is a slide about genetics. And the other thing that's in each neuronal cell are DNA, so chromosomes. So every cell of our body has the same set of DNA. So chromosomes, and then within the chromosomes, there's the genes. So we talked about that. And each gene of the GRI genes creates a different protein. So GRIN1 creates something called GLUN1, for example. And when neurons talk to each other, they do it through a genetic, or sorry, when DNA makes proteins, it does something through a genetic alphabet. So just like there's a human alphabet that makes, or human alphabet, a language alphabet that makes words, and a music kind of alphabet that makes music, there are 4 letters that make up the code to make, to basically take a gene and turn it into a protein. And depending on, and then there could be thousands of these in a gene, and depending on what the sequence is, it could make a muscle, it could make hair, it could make bone. So it's all going to depend on that sequence of letters. No, that's not true. So every gene has the exact same set of letters, and then as the body grows, there's other codes that tell it what to become. So yeah, every cell has the same set of DNA, and the most common kind of mutation in GRI disorders is missense mutation. So it's probably somewhere between 70 and 80% of the mutations that we see, and this is where there's just one single letter of these A, C, G, T that is changed, and so my son's like that. Many people are like that, and so just from we actually have 6.2 billion or 6.2 billion letters in letters in our DNA, and so for if you have a missense mutation, it's just one of those letters that's changed out of 6.2 billion, and that makes all of the difference in terms of our kids. There's also other kinds of mutations like a deletion where either one letter could be missing, or actually whole genes could be missing. And often, and there's other kinds too, like this one called frame shift, but deletions and frame shift will often result in something where the protein doesn't get made at all. So usually with a missense mutation, the protein still gets made, it just gets made kind of broken, where with these other ones, the protein doesn't get made at all. And by the way, I should note, the A, C, G, and T, what we call those letters, what they really are is chemicals. So there's 4 different chemicals that transmit, and the transmissions from those tiny little chemicals basically create us. So this is Bryson's whole exome sequencing results. And so you can see that it's GRIN1. You can see the position on the chromosome, the amino acid, the nucleotide of the amino acid. So those are some sort of the most important ones for me for learning about how Bryson's different from other people with GRIN1. And they basically both spell out the same thing. They both signify the same thing. But basically what this is saying is that in the GRIN1 gene, there's thousands of letters, and the 1,858 letter where there should be a G, there's an A instead. So it's where there should be a glycine, there's an arginine instead. And then that means that where there should be a G, no, I was wrong about that, sorry. The G to A are those letters, right? The G to A are these letters that's changed. And then under that, the G620R means that where there should be a G, there's an R instead. Okay. Loss of function and gain of function. This is something that you may have heard about. I was talking about with some families today, but for most of the GRI diagnoses, they can be, or many of them, they can be defined as either a gain of function variant or a loss of function variant. And what's interesting about these GRI disorders is that often the symptoms are the same. So whether it's a gain of function or loss of function, the symptoms can be very similar. And what gain and loss of function kind of mean is the way that the gene impairs neurons from being able to talk properly to each other. So if you think about, if you're listening to someone talking, or if you're listening to music and trying to make out the words, if the sound is very low, you won't be able to hear what the words are. If the sound is very loud, like if you turn it up to 11, then it may become distorted and you still can't hear the words. So it's something like that. So in either of those cases, the inability for one neuron to communicate properly with the other neuron results in these deficits. So those are called gain of function and loss of function. And there's many that are clear gain of function, many that are clear loss of function, but there's also a whole bunch that it's kind of hard to tell what's going on. So where there's clear gain or loss of functions, that may be indicative of the kind of medication that could be helpful. So for example, with gain of function patients, there's been several studies that have suggested that memantine, which is an Alzheimer's drug, can help to lower the volume of that music so that the neurons can hear each other and make out the words. Okay, so the path to treatments and cures. I talked earlier about how when we had this meeting in Toronto, that I really heard hope that there could be treatments and cures. And part of that reason is Dr. Amy Ramsey, who's here. When Dr. Ramsey gave a talk, actually she couldn't even be there in person. She works in Toronto, but she was in Spain, so she gave her presentation remotely. About what with the student who was in the room. But what she told us was about how she had done work where she created a mouse with a variant that could be turned off. So this mouse, grade 1 mouse, kind of grew up thinking or grew up behaving as if it had a loss of function grade 1 problem. And so it had different deficits. It wasn't as social as other mice. It wasn't able to solve mazes. It wasn't as strong. And she kind of built it with a switch that she could flick off when the mouse reached a certain age. So at that time, it was like the gene was correcting itself. And so the gene started making, instead of the broken protein, started making good proteins. And what she found is that whether she turned that gene on in the mouse's adolescence or even in the mouse's adulthood, that the mouse became a lot more like typical mice that had never had any problem. And so the hope this provided was that, even in teenagers or adults, potentially it could be possible that when we find the right treatments or cures, that there could be very profound reversal of symptoms. So that's a big part of what led us to want to found Cure GRIN. So there's different kinds of research. So basic research is really when researchers are trying to figure out, basic researchers who work in these genes and receptors are trying to kind of figure out the mechanics, like what's going on within the receptor. Not really as much about, or not necessarily about how it relates to disease, just how it behaves. And then translational is taking that basic research and applying it to treatments and cures for people. And so, that can take a couple different ways. So one part of it there is identifying potential treatments. Another part of translational is doing preclinical testing on cells or animals. And then the kind of third stage of research is clinical, which involves trials in people. And so this is all kind of the research that leads to treatments and cures. And part of what we do, I think part of where Cure GRIN has been successful is talking to basic researchers and helping them to understand the disease and making them think more towards the translational side and think about how this could apply to people, how this different kinds of medicines might work, and testing those on cells and animals. And so today we're in Chicago here for this meeting today, but then the next 2 days there are maybe 8 researchers that are coming in from I think really around the world, but especially from the Northeast, Midwest, U.S. And so I'm speaking there along with Liz Marfiash, and that's another opportunity that we have to try and convince those basic researchers to think about translational research. We also brought together a group to develop a research roadmap, and this was basically designed to identify how Cure Grid invests our resources, so time and money, but also to influence the priority of others. And as part of that, we did a survey of patients. And one of the questions we asked was whether you want us to focus on cures or treatments. So a cure being something that doesn't necessarily make people someone with a GRI disorder like a neurotypical person, but improves a number of symptoms. Where when we talk about treatments, we're thinking more of something that will just impact epilepsy or another thing or another kind of symptom. And it was pretty balanced. Like most families wanted us to focus equally on both, with a little more emphasis on the treatments than the cures. We also asked them what's the most important symptom that you want researchers to be focusing on, and intellectual disability and communication, epilepsy, and behavior and neurostorms were the three that people asked us to focus on. At the time we did this, we were only really starting to get into representing other genes, so there's a very small number of GRI patients. But you can see that even with the small number there, across all of the GRIN genes and the GRIA genes, the areas to focus on were the same. And so out of the research roadmap, we did two things. So one is kind of decided that as we were working with researchers, we wanted to focus on sort of three broad areas. So the first, when we think of a cure, it could most likely come from something that is kind of gene therapy or gene editing. And so this is where you would have a treatment that would basically go in and correct the misspelling in genes, at least in the brain, maybe in other places, and that by doing so, you could help address the deficits that are there, because like with Amy's mouse, the genes would start creating typical proteins instead of the kinds of broken proteins that are being created now. The second is drugs that specifically target the NMDA receptor or the AMPA receptor. And those could potentially be just a treatment or something that could, a treatment that affects one symptom or something that affects many symptoms. And then the third area is drugs targeting specific symptoms. So like epilepsy drugs, like the drugs that are helping people with neurostorms or paroxysmal sympathetic hyperactivity. We also came up with 10 essential questions. So I won't get into these now, but this kind of has led to a lot of what we're working with researchers on now. So 10 questions for how to get to treatments and cures. And then for each one of those, we went through a process of identifying what's the best way to get to these answers. And so we identified 4 areas where Cure GRIN can have an impact. So one is through collaboration, one is seed funding for researchers, educational opportunity. Educational opportunities could be for families, researchers, or physicians, and finally, access to patients. And then I don't have them all in here, but just as an example, one of the areas we're focused on is, one of the 10 questions is biomarkers. So biomarkers, an example of a biomarker is I had a kidney transplant a few years ago. And so I have to get my blood tested regularly to make sure that my kidney transplant is still working. My body's not rejecting it. And there's certain kinds of things in my body, so certain kinds of even nutrients that at a high level become toxic. And so those are biomarkers. Those are the first signal in the blood that the kidney is not functioning. And so we're trying to find out if there's similar things to that for GRI disorders. And if there is, it could be really helpful because, for example, if there's a treatment, it might be the first sign that a treatment's working. If we know that there's something atypical, for example, in the blood of kids with GRI, and then we give them treatments, and we can see that the blood is acting more typical. That's a signal that it could potentially also lead to more important impacts. So one of the areas here we've identified is invest in research to analyze blood and EEGs. And so earlier this year, we announced $652,000 in funding for 5 different research projects. And this first project with Dr. Maggie Caleb in New Zealand and Dr. Amy Ramsey, who I mentioned earlier in Toronto, they're looking specifically at that question around blood and biomarkers as part of the grant. And we're almost out of time, but these are some of the other projects that we're funding. And just like another quick case study, it's not just the funds that we give out, but Kieran has also been successful at working with other groups to bring more funding to them that we're not necessarily doing ourselves. So back in 2019, I went to Boston and met with a number of different biotechs, and Homology Medicines is a gene therapy company. And they agreed that if we could find a researcher who could test their medicines on mice, that they would provide them for free. And then the opportunity came up a couple of years later where Simon's Foundation was offering a grant specifically for public-private partnerships, so with an industry and a researcher working together. And so we brought this opportunity to Dr. Ramsey, who had already was working with mice and with Homology, and they agreed to come together and work on this proposal. And then it was successful, so that resulted in over $1 million for Amy's lab, plus all of the free medicine that's coming from Homology. And then a year later, I spoke with another researcher at University of California, Davis, and he was trialing a promising new kind of gene therapy for Angelman's syndrome. And I contacted him, and he agreed to kind of join the project and said he would do the work for free if we just paid for the materials. And so that expanded things. So this is the kind of thing that we try to do as well as just the direct funding that we're doing. And then the good news is that there are now a number of biotechs and industry interested in working with Pure Grin. So the first commercial clinical trial is already underway in Europe with Grin Therapeutics. They are working to get approval in the US, but they're not ready to launch that trial yet. Numora is another company they're not doing clinical trials yet, but they have us on their pipeline. They're doing preclinical work on potential treatments. NOS Pharma is actually connected with Dr. Derek Bowie at McGill University, and he has a combination therapy that he thinks will be promising for grief disorders. So I know we started late and I went overtime, but I have a cock. I'll take, if there are any questions, I'll take them before I turn things over the line.
I just have one question. One of your slides said WES results. What is that?
So that was for whole exome sequencing.
Okay.
So it's probably how most of us in the room got the diagnosis.
Thank you.
No problem. Anything else? Okay, thank you. So Lonnie, I haven't opened yours up yet, so give me a second.